we're just going to hit kind of some of the main ideas from the final exam. Some of them are just things that I've noticed are more commonly missed. But we're not going over, of course, every single topic. That would take forever. If you have any questions while we're going through this, go ahead and type them into the chat box, and I'll stop and answer them. All right, so we're going to start with important people. So our people are Anton von Leeuwenhoek, Charles Darwin, Gregor Mendel, James Watson and Francis Crick, Erwin Chargaff, Maurice Wilkins and Rosalind Franklin, Alfred Hershey and Martha Chase, and Marie Curie. And the events, they're not in order. First, confirmed that DNA was genetic material, discovered the ratios between adenine and thymine and guanine and cytosine bases in DNA, theorized about natural selection and evolution, experimented with radioactive elements, built the first microscope, discovered the methods of inheritance, discovered the structure of DNA, made X-ray crystallography images to determine the structure of DNA. So this is going to be up to you two to get these matched correctly. You can start wherever you want. If you know one, go ahead and type into the chat box which one it is that you know. Darwin, natural selection and evolution. Good. Start with the ones that you know first, and then we'll kind of whittle it down from there. Anton von Leeuwenhoek built the first microscope. Very good. Marie Curie experimented with radioactive elements. Excellent. Which other ones are we sure about? Good. Hershey and Chase confirmed that DNA was genetic material. It's the Waring blender experiment. Maurice Wilkins and Rosalind Franklin did the X-ray crystallography images to determine the structure of DNA. Great. So we have Mendel, Watson and Crick, and Chargaff. Okay, Chargaff did discover the ratios, so Mendel did something else. Chargaff discovered that there's equal amounts of adenine and thymine and guanine and cytosine, so they must pair together. So we have Mendel. Remember, he was the Austrian monk. He worked in the garden. And then we have Watson and Crick, and they made something super famous, a model of something. Mendel is methods of inheritance. Good. And then Watson and Crick were DNA. Perfect. Gotta find another color to use. Okay. And then on the next page, I have the same thing, but I have them color-coded because these lines are a little hard to see. So there they are. They're with the color that they go with. So if you need to write any of this down, I'll wait a minute and then just type done when you're ready to move on. All right, you guys did that really fast. Good job. Okay, next, writing a lab and the scientific process. Oh, good. So the scientific process, this is in order. The very first thing that you always have to do is you have to make an observation. And that, for some reason, confuses people. They always put the hypothesis first, but you have to have a question about what you're trying to learn before you can make a hypothesis. So make an observation is first. Then the next step is that you make a hypothesis. Then you conduct an experiment. You analyze the data from that experiment, and then you draw conclusions. So we have an example. So make an observation. You take notice of an occurrence and come up with a question. Then determine what you can alter about the occurrence and how you expect it to change. That's your hypothesis. And then write up a detailed procedure minimizing errors and carry out that procedure. Then look for trends between data points. That's your analysis. And then come up with explanations for the trends as your conclusion. And do you need to write any of this down, or are you ready to go on? Okay, great. That's good. That means you guys already have good notes. All right, so basic chemistry. So what we have here, this is called an atom. And our atom has 3 subatomic particles, so that's smaller than the atom, subatomic. So the first one we have is protons, and protons are located in the nucleus. And they have a positive charge. So in this picture here, they're represented by the red. They're positive, and then they're in the nucleus. Then next we have neutrons, and neutrons have no charge, and they're also located in the nucleus. So in our picture here, those are the blue ones. And then our last ones are our electrons, which are represented by the gray in our picture. And those have a negative charge and are located in orbitals outside the nucleus. All right, then we have 2 basic types of bonds. We have a covalent bond and an ionic bond. In a covalent bond, we can see that our atoms, so we have an atom here, we have an atom here, and in that one they're sharing electrons. So we can see its electron, when they share, now that electron will go around both of them. So sharing electrons is covalent. And then in ionic, they transfer electrons. So this atom here donated this electron to this atom here. So now we can see there's no electrons around that one, and there's 2 electrons around that one. And they don't change the number of protons, though. So this one, it did have 1 proton and 1 electron, which made it neutral. Now it has 1 proton and no electrons, which makes it positive. So when it donates an electron, we call that a positive ion. And then the other one accepted an electron, so it did have one positive and one negative, and now it has one positive and two negatives. So it has a overall negative charge, so we call that a negative ion. So that's the reason this is called an ionic bond, because when they donate and accept electrons, it makes them ions. And a positive and a negative ion attract like magnets. Positive attracts a negative. So that's an ionic bond. And then further on the right here, we have more covalent bonds. And when something covalent bonds together, that result, we call it a molecule. When they ionic bond, we just call it an ionic compound. So these ones here are examples of molecules. So you can see this is two hydrogens. They bond covalently, so those electrons will be going around both of them to get a hydrogen molecule. And what each atom wants to achieve is it wants to achieve a full outer orbital. So this first orbital can hold 2. So now that these electrons are going around both of them, it's like they both have 2. Here we have fluorine. They each have 7, so they each need 1 more. So they share these ones. So now it's like they each have 8, so they have a full outer orbital. And that's what all atoms want to achieve. And they can do this by either sharing or by donating and accepting. Questions on that? Sebastian? Nope. Okay. Moving on. All right, so these are some of our macromolecules. We have 4 different macromolecules. And those are, do we remember any of those? We had 4 groups of big molecules. Do you guys remember any of those? Okay, proteins are one. And proteins are made of amino acids. And proteins are also enzymes. Then our other ones, we have lipids. And those are made of fatty acids. Then we have nucleic acid. And those are made of nucleotides. Good, and carbohydrates. And last one is carbohydrates. And those are made of monosaccharides. Perfect. So then here we can see a couple of the specific ones. On the left, we have protein. We have a protein channel in this first picture, and that's embedded in the lipid bilayer, and that's where these molecules, the ones that are specific to this channel, can just go in and out, and they're going to be moving the direction of the gradient, so that's going to be going from high to low. And then on the left, these are protein carriers, which is what Mindy said. And these actually are specific to a certain substance, and the substance goes in, and then they change their shape, and they go the other direction. So in some cases, these can push them against the concentration gradient. In this picture, it's not. It's going from high to low, which is called diffusion. And this, since it's getting help, this would be called facilitated diffusion. We know it's facilitated because it's getting help, and we know it's diffusion because it's going from high concentration to low concentration. So that is that passive transport or active transport? Would these pictures here be passive or active? Passive, good, because they're going from high to low. All right, and then the picture on the right is an enzyme. The pink is an enzyme. And then in this case, this is a maltose, which is two monosaccharides joined together. So it goes into the active site on the enzyme, and then that allows the reaction to take place, which in this case is splitting those apart. And then it releases them, and once it's done, the enzyme is not used up, it's unchanged, so then that enzyme can go along and it can catalyze another reaction. So the enzyme itself is unchanged by this reaction. And do we have any questions on this one? Okay, characteristics of life. So on the left, these are the characteristics that everything that's alive has these characteristics. So they all are made of smaller units called cells. They all respond to their environment. They all grow. They are capable of reproduction. They're able to adapt to their environment. They use energy. And they maintain homeostasis, which is constant internal conditions. And then on the left, this is called the levels of organization. And this is just going from atom to organism. So the smallest unit of matter is called an atom. And then we put together many atoms to make a molecule. We put together many molecules to make a macromolecule, which we said were lipids, carbohydrates, nucleic acids, and proteins. Then macromolecules make up organelles. Then organelles make up a cell, and a cell is the smallest unit of life. So atom is the smallest unit of matter, but not until we get to a cell is that considered life. And then many cells make up tissues. Many tissues make up organs. Many organs make up an organ system. And finally, organ systems make up an organism. And then in the next class, we'll go from organism, we'll start at organism, and then we'll get even bigger up to the ecosystem. All right, so cell composition. So here we have two basic cells. We have a eukaryote, which stands for true nucleus, and we have a prokaryote, which means before nucleus. So we can see the basic difference. Our eukaryotic cells look bigger, and they look a lot more complicated. So how they're different are our eukaryotic cells. What are the two main differences? Our eukaryotic cells contain what two things that our prokaryotic cells do not? Okay, not a cell wall. Some of our eukaryotic cells don't have a cell wall, and our prokaryotic cells actually do. There's two things that eukaryotic cells, so that's plants, animals, fungi, and protists, that they have that prokaryotic cells, bacteria, do not. One is that they both have DNA, but it's one of the things that's different is where the DNA is stored. See that big black circle on the left? Good. Eukaryotic cells contain a nucleus, and they also contain membrane-bound organelles. And those do not occur in prokaryotic cells. Prokaryotes, they have DNA, but it's usually just like a circle of DNA. And they have some organelles, like we can see ribosomes in there, but they don't have membrane-bound organelles. And we can just see a few of these organelles, membrane-bound organelles, and mitochondria. What's the mitochondria responsible for? One word, starts with an E. Energy, good. We have our cell wall, so this must be not an animal cell. We have our cell membrane, which is our layer that allows some things to pass through and others things not to pass through. It's called semi-permeable. Then we have, this has a big vacuole. So because this has a cell wall and a large central vacuole, what type of cell do we think this is? Plant, animal, fungi, or protist? Plant, good. And also it has chloroplast, so that's another thing that tells us this is a plant cell. And chloroplasts take energy from the sun and convert it into sugar. All right, looking at cell size, so we spent quite a bit of time on this in Unit 2. So we know that we want to have a large ratio of surface area to volume, because as our cell gets bigger, the surface area, which is the cell membrane, doesn't grow proportionally. So we end up with a cell that's too big, and so it can't transport nutrients all the way across the cell. So it can't transport nutrients all the way across the cell, and it can't transport wastes all the way out of the cell. So what we want is a small cell with a large surface area to volume ratio. And when our cells get too big, they divide. Do we have any questions on that? So that's what our potato lab was about. Okay, cell transport. We have two types, passive and active. Passive does not require energy. Active does require energy, and we can see that energy right there in the form of ATP. So for passive transport, we have three types. Well, we have diffusion, which is where small molecules are able to diffuse straight through the membrane. We have osmosis, which is the same as diffusion, but it's specifically for water molecules. They move from a high concentration to a low concentration. So same as diffusion, but with water, is called osmosis. And then we have facilitated diffusion, which is diffusion, but it needs a helper. And those helpers can be either our protein channels or our protein carriers. But they're passive transport because they go from high concentration to low concentration. So it's moving along the concentration gradient. And then active transport does require energy. So there's our ATP. And the reason it needs energy is because it's pushing substances against the concentration gradient. We can see there's less squares here. There's more squares here, and it's pushing them to the direction there's already more. So it's going against the concentration gradient, and that takes energy. So picture if you had something that's way too full, your suitcase that's way too full, if you were to open it and try to push more stuff into it, that's going to take energy. It's going to take some muscle. And the specific one that it's showing us here is the potassium-sodium pump. And then other types of active transport are endocytosis and exocytosis. So that's where the cell engulfs either a large molecule or a lot of small molecules and pulls it into the cell, or opposite, a vesicle moves to the cell membrane and it releases it to the outside of the cell. Does this sound familiar? Do we have any questions about this topic? Okay. All right, cellular respiration and photosynthesis. So we have cellular respiration occurs in the mitochondria. And that's the reason it has all those folds. It gives more surface area for this reaction to take place on. And cellular respiration takes glucose and oxygen, and it converts it into water and carbon dioxide, and it releases energy. So this is how heterotrophic organisms get their energy. And then also autotrophic organisms do this like when there's not sunlight available. So cellular respiration, we go from C6H12O6, which is glucose, plus oxygen, and we produce carbon dioxide. So think when you breathe out, carbon dioxide comes out, and also water vapor. If you breathe out on your hand, you can feel that it's wet. You've got water vapor, and then it makes energy. And then the opposite of this process is photosynthesis. Photosynthesis occurs in the chloroplasts, and it requires sunlight. So we can see here sunlight is entering the ecosystem, and it's the opposite of cellular respiration. It requires oxygen and carbon dioxide, and then in the presence of sunlight, it turns that oxygen and carbon dioxide, or excuse me, water and carbon dioxide into glucose and oxygen. So you can see 6 carbon dioxides plus 6 waters, and then we have to have energy put into it. That's our sunlight. Produces a molecule of glucose and 6 molecules of oxygen. Cell division. So we talked about 2 different types of cell division. We had mitosis, which is how all of our cells except for our sex cells divide, so all of our body cells, and we call body cells somatic cells. Soma means body. And in mitosis, we start with 1 parent cell, and then we end up with 2 daughter cells that are identical to each other and are identical to the parent cell. And in the cell cycle, we have interphase, and then goes into mitosis. We have prophase, metaphase, anaphase, telophase, and then cytokinesis. So we start with 1, and we end up with 2 that are identical to each other and identical to the parent. And then meiosis is different because it divides twice, and it leaves us with 4 gametes that they're all different from each other, and they have half of the amount of genetic information as the parent cell. And the reason we want it to be half is because, say this is an egg cell, it has half the amount of genetic information, and then it'll join with the sperm cell, which has half the amount of genetic information. So we'll end up with a zygote that has a full set of information. So meiosis only occurs in our gametes, which are our sex cells. You have any questions on this part? Alrighty. So we said here's a cell cycle. So the majority of it is interphase. It's getting ready to divide because every cell is somewhere on this in the cell cycle at all times. Then we have a couple of checkpoints, and these are points where if things aren't going kind of according to plan, if the cell hasn't grown big enough, or if there's been errors in the DNA replication, that it can either wait and it can go through that again until it's ready, or it can signal cell death. So interphase is G1. Our cell grows. and checkpoint. The S phase, that's when our DNA replicates. And then G2, it continues to grow and it prepares for division. We have another checkpoint. And then we go into mitosis. So prophase, telophase, anaphase. And remember, anaphase is our most important to the cell having the right amount of the correct genetic information, because if they split incorrectly, anaphase away, they're moving away from each other. If they split incorrectly, then we could have some mutations. We could have some cells that don't have the correct amount of genetic information in them. And then telophase and then cytokinesis is the division of the cytoplasm. Mitosis is the division of the nucleus. Cytokinesis, division of the cytoplasm. All right, DNA versus RNA. So which one of these is DNA, the left or the right? The right. Good. And how did you know that? Okay, because DNA has thymine. Good. And we have a few differences between them. So DNA is double-stranded. It contains thymine. It has deoxyribose as the sugar. And it is stuck in the nucleus. It can't leave the nucleus. Okay, so those are some differences from RNA. RNA is single-stranded. It contains uracil as one of the nitrogen bases. It has ribose as the sugar. And it travels from the nucleus to the ribosome. And I know that transcription and translation was a little tough, so we're going to do practice with that. All right, so it wants us to transcribe this gene. So transcription is DNA to mRNA, which stands for messenger RNA. It has to give the message from the DNA to the RNA so that the RNA can carry that message to the ribosome. So let's transcribe it. Okay, so T pairs with A pairs with U, C pairs with G pairs with C, another C, T pairs with A, U, G, G, U, C, A, C. Great job, Sebastian. Do we have any questions on that? Denise, do we still remember how to do that? Okay. And then next is to translate the gene. So that's where we're going to go from mRNA to amino acids. Okay, so what is AUG? Good. Methionine. And that's also the start codon. Then next, CCA. Proline. Next one. Good. Tryptophan. So that was UGG. And then we had UCA. Good, which is serine. And then we had just one left over that I just kind of picked a little chunk of one. Okay. And so what it transcribes, it gives the message to mRNA, so the DNA temporarily unzips. And how the RNA gets the message is through base coding or base pairing. T pairs with A pairs with a U and RNA, and C pairs with G and G pairs with C. And then that piece of RNA goes to the ribosome. And at the ribosome, tRNAs, which stands for transfer RNA, or I like to call them taxi RNA, goes and picks up the amino acids, and then it comes back to the ribosome, and it knows where to drop them off because of base pairing. A to U, C to G. So that's translation, because we're switching languages. We're going from nucleic acid language to protein language, which is amino acids. All right, biotechnology tools. So just going over a couple of them. Gel electrophoresis, and that's what we're showing in the picture here. Exposes charged molecules like proteins and nucleic acids to an electric field that allows them to be separated by size. Because it's in an electric field, the ones that are shorter are going to travel further, and the longer are not going to travel as far. So we end up with them separated based on size. Then PCR, which stands for polymerase chain reaction, is a technique that helps scientists make copies of key portions of DNA. The results of PCR can be put through an electrophoresis and analyzed for the presence of DNA bands of a certain size. So the PCR makes it so scientists can make a bunch of copies, because if you have just a DNA sample, you want to make a bunch of copies of it so that you can run various tests on it. Sanger sequencing is a technique for determining the complete sequence of bases for a particular piece of DNA. And I'm not going to be able to show the video through Adobe, so I'm going to put the link in the chat box. Well, I say I am. So let me get the link for you, and then go ahead and watch the video and let me know when you're done. All right, great. And then last we have our DNA barcoding, and this is used to identify a species. Okay, so a little bit more. So here we have a picture of a DNA amplification, and DNA amplification, sorry, I'm going to type on it. Okay, it artificially increases the number of copies of a particular DNA fragment into millions of copies. And we do this through PCR. Then nuclear transplantation is exactly what it sounds like. You're putting a nucleus into something else. So it's the nucleus of a donor cell is relocated into a target cell that has the nucleus removed. And so this is also called nuclear cloning. And then genetic modification is the direct manipulation of an organism's genome and a good example of this is when we did GMOs. They can transfer a gene within and across species to produce improved or novel genes. So that's like how they put the keratin gene from carrots into rice to make golden rice. And are you ready to move on, or are you writing this down? Perfect. Okay, so here's some more biotechnology terms, and these are matched up. They don't have to be matched. They're just straight across. So reproductive cloning is creating a clone of an organism that is born. So that would be like Dolly. Therapeutic cloning is creating clones of certain cells for medicinal purposes. So this is when like they will grow cells in like a petri dish. So maybe they're doing like a skin graft or something. Stem cells are special cells that can change into any type of cell. So they haven't differentiated yet. They're able to change into other cells. Regenerative medicine is a practice that uses stem cells to create new organs to replace old ones. Genetic engineering is modifying the genes of an organism. Recombinant DNA is DNA that contains genes from more than one organism. So if we were to look at the DNA from that golden rice, it's recombinant DNA because it has genes from carrots. A vector is a modified plasmid into which desired genes are inserted. And the plasmid is circular DNA. Gene cloning is making copies of a specific gene. Plasmid is a circular DNA in bacteria, and this plasmid is what makes them resist antibiotics. And then a transgene is a gene that was taken from one organism and inserted into another. So let me know when you're ready to move on. We're almost done. Okay. And then last, the Human Genome Project. And this was the project that determined the sequence of the entire human genome found within an organism, the cell of an organism. So it identified the genes, like our whole genome, and along with this, it identified a lot of the genes that cause diseases. It helped that we know now about 1,800 of the genes that cause genetic diseases. There was a discussion post where it asked you about a genetic disease and it asked what chromosome the mutation was on that caused it. So, I mean, we can pinpoint it not even just down to the chromosome, but down to the exact gene that causes the disease. The Human Genome Project allowed the creation of PCR-based tests for diseases. So an example is we now have like a routine test for genetic forms of breast cancer. So if you know if it runs in your family, it's something you can be tested for and see if your likelihood of getting that genetic form of breast cancer. It allows for the development of technologies to treat these disorders. So we said it identified different genes and where they're located on our chromosomes. We can actually say which chromosome it is, which arm of the chromosome, and which gene it is, which is pretty crazy. And also it discovered that humans have about 20,000 genes, and that's a lot less than they previously thought. Because we look at genes in other organisms, we just figured that humans are the most advanced organism, so we must have the most. And that's not quite how it works. So 20,000 genes was much less than we thought. But there's so much technology that's being developed based off of knowing this entire human genome. So it's a pretty incredible discovery. And this is also what all of those tests, so it's like ancestry tests and stuff, that's what all of that is based off of. And that's the end of what I have for you. So here I just have some test-taking tips. Multiple choice questions. They shouldn't be tricky. Let's not let them seem tricky. So first, read the question before you look at the answers. And read the question and come up with an answer in your head. And then look at the choices so that you don't look at the choices and it throws you off. Go ahead and eliminate the answers that you know aren't right. Read all of the choices before choosing. There isn't a guessing penalty, so do not leave questions blank. If you're not sure, make an educated guess. Do not leave it blank. In the all of the above or none of the above choices, if you're certain that one of the statements is true, don't pick none of the above. Or if you're sure that one of the statements is false, don't pick all of the above. Those ones can be a little bit tricky. Make sure you're taking your time on your test. Remember, the tests aren't timed as long as you're consistently working on them. If you get up and leave, when you come back, the test will be closed. But if you're working on it, it's not going to time you out. So take your time. And that's it. Good luck. Do you guys feel like you're ready for your final exam? Awesome. So